Narcolepsy is a chronic neurological disorder characterized by excessive daytime sleepiness, EDS, and a range of other symptoms, including cataplexy, sleep paralysis, hypnagogic hallucinations, and disrupted nighttime sleep. This disorder affects approximately 1 in 2,000 individuals, with a higher prevalence in certain populations. This script aims to provide a detailed understanding of the complex mechanisms underlying narcolepsy, including neurotransmitter dysregulation, hypocretin deficiency, role of orexin receptors, HLA associations, autoimmune mechanisms, genetic risk factors, neural circuitry, sleep architecture, diagnostic criteria, excessive daytime sleepiness, cataplexy, and management and treatment options. Narcolepsy involves dysregulation of several neurotransmitters, particularly the monoamines. Research has shown alterations in the levels of neurotransmitters such as dopamine, norepinephrine, and serotonin in individuals with narcolepsy. These imbalances contribute to the symptoms experienced, including EDS and disrupted sleep patterns. Further studies are needed to fully comprehend the specific mechanisms responsible for these dysregulations. One of the key findings in narcolepsy is the deficiency of hypocretin, also known as orexin, a neuropeptide produced in the hypothalamus. Hypocretin plays a crucial role in regulating wakefulness and maintaining a stable sleep-wake cycle. In individuals with narcolepsy, there is a significant reduction in hypocretin levels, leading to the disruption of normal sleep architecture and the manifestation of symptoms such as EDS and cataplexy. Orexin receptors are the targets of hypocretin in the brain. There are two types of orexin receptors, OX1 and OX2. Research has shown that the loss of functional orexin receptors is associated with the development of narcolepsy. These receptors are involved in the regulation of various physiological processes, including arousal, feeding behavior, and the maintenance of wakefulness. Understanding the role of orexin receptors is essential for developing targeted therapies for narcolepsy. Narcolepsy has a strong genetic component, and certain variants of the human leukocyte antigen, HLA, genes are strongly associated with an increased risk of developing the disorder. Specifically, the HLA DQB1x602 allele is present in a majority of narcolepsy cases. The specific mechanisms by which these HLA variants contribute to the development of narcolepsy are still under investigation, but it is believed to involve an autoimmune response targeting the hypocretin-producing cells. Increasing evidence suggests that narcolepsy may be an autoimmune disorder. The destruction of hypocretin-producing cells in the hypothalamus is believed to involve an immune-mediated process. Studies have shown the presence of autoantibodies targeting hypocretin receptors and other proteins involved in the function of these cells. The exact triggers and mechanisms of the autoimmune response in narcolepsy are still being investigated, providing insights into potential therapeutic targets. Apart from the HLA associations mentioned earlier, other genetic factors contribute to the risk of developing narcolepsy. Genome-wide association studies have identified several genes associated with narcolepsy susceptibility. These genes are involved in immune regulation, neuronal development, and neurotransmitter signaling pathways. Understanding these genetic risk factors can enhance our understanding of the disorder's pathogenesis and aid in the development of personalized treatments. Narcolepsy involves the dysregulation of neural circuits that control sleep-wake transitions. Various regions of the brain, including the hypothalamus, thalamus, and brainstem, play crucial roles in modulating sleep and wakefulness. Disruptions in the neural pathways and interactions between these regions contribute to the abnormal sleep patterns and symptoms observed in narcolepsy. Further research is needed to fully elucidate the precise neural circuitry affected in narcolepsy. Narcolepsy significantly alters the normal sleep architecture. Polysomnography studies have revealed characteristic abnormalities, such as shortened sleep latency, rapid eye movement, REM, sleep occurring early in the sleep cycle, and frequent awakenings throughout the night. The disruption of sleep architecture contributes to the excessive daytime sleepiness experienced by individuals with narcolepsy. A comprehensive understanding of these alterations can aid in diagnosis and treatment approaches. The diagnosis of narcolepsy involves a combination of clinical evaluation, subjective and objective sleep assessments, and laboratory testing. The International Classification of Sleep Disorders, ICSD, provides specific criteria for diagnosing narcolepsy, including the presence of excessive daytime sleepiness and cataplexy, as well as specific polysomnographic findings and the presence of hypocretin deficiency. Accurate diagnosis is essential for appropriate management and treatment planning. Excessive daytime sleepiness is the hallmark symptom of narcolepsy. Individuals with narcolepsy experience persistent sleepiness that is often unrelieved by sufficient nighttime sleep. This excessive sleepiness can significantly impact daily functioning and quality of life. Various factors, including neurotransmitter dysregulation, hypocretin deficiency, and disrupted sleep architecture, contribute to the excessive daytime sleepiness observed in narcolepsy. Cataplexy is a sudden loss of muscle tone triggered by emotions, such as laughter, anger, or surprise. It is a cardinal symptom of narcolepsy with cataplexy. Cataplexy occurs due to the loss of muscle tone during REM sleep intruding into wakefulness. The specific mechanisms underlying cataplexy are not fully understood, but it is believed to involve the dysregulation of neural circuits and impaired OREXIN signaling. Treatment options for cataplexy aim to minimize its impact on daily activities. Narcolepsy is a chronic condition with no cure, but there are various strategies to manage its symptoms and improve quality of life. Treatment options include pharmacotherapy, lifestyle modifications, and behavioral interventions. Stimulant medications, such as modafinil, and selective serotonin reuptake inhibitors, SSRIs, are commonly used to alleviate excessive sleepiness and cataplexy. Additionally, establishing regular sleep schedules, incorporating scheduled naps, and addressing comorbid conditions are important aspects of disease management. In conclusion, narcolepsy is a complex neurological disorder involving a range of molecular, genetic, immune, and neural factors. Understanding the mechanisms underlying narcolepsy can improve diagnosis, treatment options, and overall management of this debilitating condition. Ongoing research continues to shed light on the intricate interactions between neurotransmitter dysregulation, hypocretin deficiency, genetic risk factors, and autoimmune processes, paving the way for targeted therapies and improved patient outcomes.